All right, folks, we are through the worst part of cell energy. The worst part is cellular respiration. It's by far the most complicated process of the energetic reactions. Photosynthesis, I think you're going to find to be a little bit easier. In the light and pigments and chloroplast video that you watched, I went over some things that you're going to need to understand this particular video. So if you have not watched that, I would recommend that you stop this video and go back and watch that before you watch this, because that's kind of background for understanding photosynthesis. So photosynthesis is an autotrophic process to convert light energy into chemical energy in the form of carbohydrates. So where respiration, it takes chemical energy from carbohydrates and converts it to chemical energy in ATP. This process is actually going to be the step that occurs before that in autotrophs, where you're making the carbohydrates themselves. Just like respiration, this is a redox process, sometimes not necessarily because electrons are stripped away, but because they're moved closer to one atom and further away from another. And this is done exclusively in the thylakoids of the chloroplast, in the chloroplast. And there are two sections of that. The thylakoids are where the first stage of photosynthesis is going to occur, and the stroma is where the second stage is going to occur. Okay, so if you're looking at this is the first stage right here, number one. The light-dependent reactions, also known as the light reactions, these are going to take place on the thylakoid membrane, and then the second step, the Calvin cycle, is going to take place in the stroma. Okay, so two-step process for photosynthesis. Now, we're going to focus exclusively in this video, for the most part, on the light reaction. So I'm just kind of mentioning the Calvin cycle here. We'll get into that in the next video. Just like photosynthesis, we're going to break this up a little bit just to let things marinate and digest for you. All right, so light reactions should be a fairly familiar looking process to you because they involve an electron transport chain. So just like the electron transport chain you saw in cellular respiration, you're going to see an electron transport chain here. The whole process starts out with something called photolysis. Photo, of course, means light. Lysis means to split. This is the energy from light splitting water. Okay, that's photolysis. So that's going to be the first step. So a word about these reactions first before we get started. Let's start with where they take place. Again, this is on the thylakoid membrane. And I mentioned that this is going to start with photolysis of water. So if you're thinking of that photosynthesis equation, water is going to come in, right? All right, so a word about what happens when light strikes something, okay? You can have fluorescence, and fluorescence happens more often than you think. It's not the best thing, but essentially you have a molecule of chlorophyll that gets an electron excited, but the intensity or the energy of the light is not enough to raise that electron high enough to cause the reaction that we want to see. And in this case, it's photosynthesis. Photosynthesis is going to involve this and this. I always abbreviate photosynthesis, PS, by the way. Photosynthesis involves resonance, where electrons get excited, and then the energy from that electron bounces to the next pigment molecule and to the next and to the next and to the next until they reach a reaction center. And at that reaction center, we would see that electron get excited to the point that it's going to reduce something. It's going to be accepted by some electron acceptor and get reduced. And what you're going to see is that the pigment at the reaction center is going to get oxidized. Okay, so that's where you're going to see reduction oxidation. But oftentimes in photosynthesis, you're going to see a little bit of this. You're going to see electron energy being transferred to neighboring pigment molecules. And then once those that energy, once it resonates to that reaction center, that's where you're going to see the light reactions kicking off, and they kick off with this reduction and oxidation. Now, like I said, fluorescence happens. It happens all the time where, some of the electrons start to get excited, but then they just kind of drop back down. And when they do, they give off fluorescence. A fluorescent, it's kind of a form of light, I guess, but that can happen. It's a little bit inefficient, but it is what it is. But NASA scientists are actually using that, since it is a byproduct of photosynthesis, to measure using images from satellites that pick up fluorescence to see the rate of photosynthesis around the world, which is pretty cool. All right, so there is that. All right, let's go back to the light reaction slide. Okay, I know there's a lot here. It looks big and scary. What you must understand is that you have two reaction centers in the light reactions, okay? And a reaction center is basically a complex of light harvesting pigment molecules and then their associated proteins. And within the reaction center is a pair of chlorophyll molecules called the P680 chlorophyll molecules within this photosystem 2. And then within photosystem 1, that pair of chlorophyll molecules is called P700. And those are the chlorophyll molecules that are actually going to cause the reduction or oxidation. They're going to kick off the actual electron transport chain that is the light reactions. Okay, so that's what you need to know there. The reason why they're named this is that the P680 chlorophyll molecules get excited, their electrons are excited to a higher energy state at 680 nanometers of light, and then the P700 is at 700 nanometers of light. These photosystems are listed on this slide in the order in which they occur. The reason why they're named the way that they are, because you're thinking, okay, why is photosystem 2 before photosystem 1? They were named in the order in which they were discovered. So photosystem 1 was actually discovered prior to photosystem 2. All right, so essentially what happens is a photon of light is going to strike water, and that's when photolysis is going to occur. And that energy from the photon of light is literally going to shake that water molecule apart. Oxygen gas is going to go spinning away at this point. You're going to end up with a buildup of hydrogen ions. That ought to sound familiar to you, right? Because as those hydrogen ions get shaken away from water, their electrons get stripped away and excited to high energy states. Okay, and that's what's going to start this. So essentially, you get light striking a leaf, you get resonance where electrons get excited in neighboring pigment molecules until you reach this special photosystem 2. And at photosystem 2, that's where photolysis is going to occur. And when photolysis occurs there, the electrons from water are going to get passed to the P680 pair. And the P680 pair is always in an electron deficit, okay? Because when those electrons are passed to the P680 pair, they're going to get elevated to a higher energy state, and that's where you're going to get reduction and oxidation, okay? So they're going to reduce NADPH or something else, depending on whether you're talking about photosystem 2 or photosystem 1. But you're going to reduce NADPH at the very end. All right, so let's take a look. Oh, by the way, let's just fill out in our chart. Remember, the water comes out as O2, oxygen gas. Okay. All right, so here we go. So here's our photons of light. You can see the photons strike, and then you do get some resonance, right? So some of the light is striking complexes that are not at photosystem 2, but presumably those electrons or the energy is being passed to this P680 pair. And notice here's photolysis. So 2 water molecules get split apart. You end up with 4 hydrogen ions, 4 electrons, and these are what's going to get passed along our electron transport chain. And then, of course, oxygen gas, which is a waste product. Okay, so we don't need that for photosynthesis. That's a waste product. So the electrons here end up being passed down the electron transport chain. And just like you see in any electron transport chain, as they bounce along, they lose some of their energy, except that when they reach the P700 pair, they get re-excited by more light. And when they're re-excited, they're going to get passed along again until they can reduce this NADP plus. And I always remember that this is NADP plus, not NAD plus, because NADP plus is in photosynthesis. Okay. The other one is the cellular respiration one, but the one that's NADP plus is in photosynthesis. And that's going to produce for us electron carriers. You know what those are. Okay. So that's the light reactions in a nutshell. Let's go back now and fill out our what we have so far. And then we've got to talk about what's going on in the background, right? Okay, so we're also going to bring two NADP plus to the party. And they're going to come off as two NADPH, right? Okay. So they're coming off here. We've got, electron carriers that are being put together. Now, obviously, the hydrogens for this NADPH, just like in the light reactions of respiration, they're coming back. These are from right there, from the water molecule. But what you don't see here is one other thing being produced. When we do electron transport chains, right, we strip the electrons away from hydrogen. We build up hydrogen on one side of a membrane. As a result, you get chemiosmosis, right? You get that proton motor force, right? Because those H pluses that you see here are protons, right? So those protons or H pluses are going to travel through an ATP synthase molecule, and you're also going to create ATP here, okay? So yay, we've done all this, right? Yay, we've passed electrons around and we've made some electron carriers, yippee, right? But the big major thing that's really important here, and it's very much going on in the background, is that we're going to create ATP. We're going to create some energy from this process. Okay, now notice I'm not paying attention to how many in this process. It's not the same as cellular respiration where we're comparing efficiency like we did between aerobic and anaerobic. Now we don't need to worry about that. Now we just know ATP is formed. All right, so the other thing that's going to go in is ADP, and it's going to be released as ATP. Now, why is this important? We've got electron carriers for building sugar. Later, right? In the next step, we're going to actually build the sugar. But the other important thing here is this guy. We've converted light energy to chemical energy. And that's the important thing. This first step, these light-dependent reactions, are all about taking sunlight energy and now converting it into an energy form that now the cell can take and use to form bonds between other chemicals later. Later on, we're going to take this ATP and we're going to hydrolyze it. And we're going to use the energy that's released from the hydrolysis of ATP for building our sugar molecules that we're going to create in the last step of photosynthesis. One other thing I forgot to mention to you here, just as a point of interest. I don't know that the College Board is going to necessarily mention this, but just so that you understand that there are some adaptations that organisms can make. There is an alternate path that electrons can take, and it's called cyclic electron flow. And this is because some organisms, certain bacteria, don't have photosystem 2, and so they only have photosystem 1. What this looks like is this. So photosystem 1 receives photons of light, resonant energy from neighboring pigment molecules. That electron is excited. It gets passed down an electron transport chain, right? And again, ATP via a proton motive force is going to happen there, but then the electron gets passed back to the P700 to get re-energized, okay? So that's going to happen there as well. So in cases where you don't have that second photosystem, some organisms use a different method for passing electrons.